Ticks. They're a scourge on humanity. They're vectors of Lyme disease, Rocky Mountain spotted fever, anaplasmosis, and other pathogens that can be debilitating and even deadly. And they're marvelously adept at going unnoticed when latching onto their hosts. Having been blessed by the devil, perhaps, with an anesthetic and inflammation-mediating saliva that allows the tick to bury its evil mouth parts into the flesh of its host without causing pain. I live in rural Northwest Pennsylvania where ticks are abundant, and despite our best efforts to keep them off of our bodies, everyone in my family has been victimized by the little parasites. My son was even diagnosed with Lyme disease a few years back after a tick bite went unnoticed until the telltale bullseye rash associated with Lyme appeared on his inner thigh. I'm Matt Pillar. This is the Business of Biotech, and I'm done ranting about how incredibly nasty ticks are, at least for now. In fact, my guest on today's show is going to instead flip the script and show us how the incredible function of nomacopan, the protein responsible for the inflammation mediating properties in tick saliva that lets their bites go undetected, might actually lead to promising anti-inflammatory therapeutics. That guest is Rachelle Jacques, President and CEO at Akari Therapeutics, and she's with me in the studio today. Rachelle, welcome to the show.
Thanks, Matt. So happy to be here.
Rachelle, you were turning red as I was expressing my disdain for ticks. You're saying, no. We're developing cool things from this evil tick saliva.
We will definitely flip the script. You're right.
Glad to hear it. All right, so we're going to get to that. We're gonna learn all about Akari and its ticks shortly. But first, I want to get to know you a little bit better. Looking at your resume, the first 7 companies you worked for spanning the mid to late 90s into about 2019 include Ernst & Young, Deloitte & Touche, Dow Corning, Baxter International, Baxalta, Shire, and Alexion. And if you look at those, you're looking at the first 7 and they're like multi-billion-dollar businesses back to back to back. Then you make the stark move into new and emerging with your first CEO role with a decidedly small startup, Enzivant. So tell me about that. Give me the backstory on this drastic shift from moving from one, Fortune whatever company to the next, and then on to Enzivant.
Sure. It does look drastic, but I will, say that the CEO role has kind of always been in my ambitions, and I think primarily because of the challenge that is presented in these roles, absolutely. And I think Enzivant was particularly special on this front, where the stakes were very high and the challenges were clear. Well, the regenerative medicine space. What we were doing there is trying to get a novel one-time tissue-based therapy to through the FDA approval process and to children who have a condition that is uniformly fatal. So stakes were high. The challenges were real. Of course, the CMC manufacturing challenges and regenerative medicine alone can definitely keep you up at night.
Yeah.
But the great thing is, it was one of those missions where the outcome of that could be incredibly impactful. And I remember when I was looking at the opportunity to join Enzivant originally, and there was a moment when it became very clear to me that I wanted to lead that mission. And that was, I saw the Kaplan-Meier survival curve for the children who had been treated. And again, this is a uniformly fatal condition. And that survival curve for the kids who've been treated in the clinical studies was, I'd say, nothing short of remarkable. I'd never seen anything like it. I still haven't seen anything like it since. And so I knew I needed to do it. And what was great is, having an excellent team that was absolutely committed to the mission. Failure wasn't an option. And so all of those things are the things that, someone really, I think, can get up every morning and be, energized and challenged by. So it was great. It's perfect for me. And definitely the, really, I'd say maybe the best moment in my career, getting that through an FDA approval and knowing that we could offer that to the children. So it was a pretty amazing experience. And so, yes, while it seems like a stark change, it had all of the things that I would have hoped for.
Yeah, and that's awesome. I mean, obviously you're not the first emerging biotech CEO that I've talked to who has come out of, you know, the big side of the business.
Yeah.
But and it's always interesting. I like to ask the questions. Usually I ask, you know, how those big experiences prepared you. And I want to know that, but I want to flip it around, and I want to start with like the more maybe the more compelling question around like when you realized that stark difference when you came out of the big you know, these big multi-billion-dollar companies and went to work for Enzapont. What surprised you, caught you off guard? What were you maybe least prepared for?
Least prepared for. It's a great question. I think that it's well known that resources are different.
Yeah.
So that's kind of a, you know, an expectation, even though when you know something intellectually and you experience it, it can be quite different. You know, facing those kind of trade-off decisions and deciding where to put your priorities and where to focus the resources, because, you know, in a small company, we never have everything we need or want. So that's, you know, that was definitely a challenge. You know, probably for me, the bigger learning or the bigger kind of, you know, aha moment is really in realizing that there's a lot that we all bring with us from one experience to the next, and you can't really, you know, lose yourself, even though it seems like a very different kind of a world. Okay, I'm moving from a large biopharma into a small biotech. You know, things should be different here, and maybe I need to be a bit different here. But in fact, you know, all of the experiences that lead up to that, they're all valuable. And so, you know, for me, it's a bit of, you know, don't forget, you know, what you bring to the table, and don't lose yourself as you kind of enter the new into the new realm. And I've experienced a bit of that in the past, as well as, you know, even in large companies. I was out at calling the outposts from time to time.
Mm-hmm.
And you do have that kind of same experience of, it's a very different environment, can be a very different culture. And you're working in the headquarters versus going to Shanghai and running an emerging business, very different.
Yeah.
But, you know, you have to bring those skills with you. And, you know, I remember over the years, especially culturally, really trying to adjust myself and fit in and not be that American. And you realize, gosh, you know, that I'm kind of also not bringing some of the skills to the table that are actually necessary here.
Yeah.
So yeah, it's a very interesting experience, and there's definitely a lot of learning in those transitions.
Yeah. It's fine. I like the way you put it, the you know, the intellectual awareness versus the reality of the situation. What is it about the human brain? Is it just like optimism and naivete that, you know, hey, I know what I'm getting into, but I don't really feel the pain until I'm into it, right?
You know, I do always believe whenever you're going in a new situation, you think you have it figured out what's different, and you think you have it figured out, you know, well, I know what I'll learn in this next experience, but the fact is you really don't. And it's not until you get in there and you realize, oh, okay, so the high level view of it is, oh, gee, we have fewer resources. But when you get into what does that mean, and what kind of decisions need to be made, and where do we need to be comfortable with taking risk, and how do we think about, you know, what's absolutely necessary now versus later, every single one of those daily decisions are really shaped by that reality. And I think that's where we don't quite get it until we're in the moment.
Yeah.
And we're, you know, in the day to day. So you get it from a high level conceptual, but how it actually plays out day to day can be I think eye-opening.
Yeah. Do you have any advice, real quick on this? Do you have any specific advice for like the thing to do in the moment when you're facing one of those realizations, right? So you made this transition, like we've ascertained, you go in horns down and ready to fight, and then you lift your head up one day and you're like, oh boy, I didn't anticipate this, or I didn't realize it was going to be this stressful, whatever it might be, whether it's a resource, you know, an HR, you know, the fact that you're working in such, you know, a smaller team. Any advice for like the thing to do in that moment, whether it's stop, take a breath, you know, take a break, take a walk. I don't know.
I have two
what is Rachelle?
Conflicting things going on in my head right now. On one hand, I want to say, you know, go all in. If you're gonna make a decision, and that's a path you've decided to take, then take it and execute. Make sure that you execute fully, and that you've bought into that path. Although while I'm saying that, I have in my mind, gosh, there are a lot of times you have to be flexible. So, kind of, and how do you do that? I don't know. As new information comes, I think we always have to be willing to say, is there a pivot here? Mm-hmm. And so while you're all in 100% engaged in executing the thing that you've kind of set out as the priority, I think it's really important that we still, especially in early stage companies, we still stay open to those pieces of new information that might reshape things.
Yep. Yeah. All right. I could sit here and ask you philosophical questions about how you, how you deal with the daily nuances of being a biotech leader, but we'll get back to Akari real quick. What were the you joined earlier this year? What were the circumstances that led you there?
Yeah. So I joined Akari 6 months ago. And, but it's not my first time in the complement space. So it is a, bit like coming home into complement. I had been previously with, one of those companies you mentioned is a complement company that really has established the science around that and the promise of complement inhibition. And I loved it at that time. I think there's a ton of possibility in this space. So, coming into Akari, what I really liked is, we have a bispecific as our lead asset. And so not only are we looking at what can we do in terms of complement inhibition, but also, we have another mechanism of action in our asset, which is another important modulator of inflammation. So I really see that as an opportunity to potentially address a number of disease areas that aren't currently being addressed by complement inhibition. So I think it's an exciting possibility. I think there's a pretty common belief that there's a lot to be done still in terms of reaching more patients with this kind of technology. And so it's great to be part of that and to be able to drive it with a bit of a different, a different offering, a little innovation on the complement only inhibitors.
Yeah, good deal. So I went long on my rambling diatribe about how much I hate ticks, and I want to I want to get into what makes Akari tick, and I mean that pun sincerely. The pun is absolutely intended.
Good.
So I want to start with a backstory on that. Your CSO discovered this protein, as I understand it. So tell us about that. Tell us the story and how the tick was identified as a source of a potentially therapeutic protein. Then we'll get into, the science behind what Akari is doing with this discovery. But I kind of want to hear the origin story. Like, how are we? The first time you and I talked, I'm like, I'm envisioning these scientists in a wet lab, like trying to milk a tick.
Right.
What's going on there? How'd that come about?
How did this happen? Right. Yeah. So despite your disdain for ticks, and I have to admit, I'm in Massachusetts, so I think we probably share some of the tick challenges, but this is a little bit different. So yeah,
I could go on. I mean, we could spend a lot of time talking about this, which I don't want to do. I've already made my my sentiment known. But honestly, I'm 47 years old, and when I was a child, this wasn't an issue. We didn't worry about ticks. We didn't spray down for ticks. Like it's seriously like it's I don't know if it's climate change or what it is, but areas of the country that back then were unaffected are entirely consumed at this point. So yeah, there's plenty of resource to make discoveries around what ticks can and can't.
There sure is. So we might as well make something good of it, right? So as you mentioned, our chief scientific officer, Miles Nunn, is the person who discovered the protein. And he'll say it's a bit of serendipity, really. He was working in a lab where they were studying exactly what you're talking about, these tick-borne diseases, their transmission and so on. And there was a kind of a subset in the lab that became interested in, really understanding how the ticks modulate the host responses to their own benefits. And in your opening, you talked about some of these things, but how is it that when a tick feeds, you don't feel it? Mm-hmm. And you don't groom it away, and there's not the inflammation, there's not pain, there's not itch. why is that? And so this team really wanted to understand that better. And, when ticks feed, it's actually their saliva that carries a number of proteins, actually thousands of proteins. And something there seemed to be responsible. So that team worked to try to isolate those proteins, which you can imagine was a pretty complex and manual process to get to the tick's saliva and to extract the proteins and isolate those and study them. But that's what they went about doing. And as a result, they got to a point eventually where they had kind of a subset of proteins, just a handful, if you will, that they thought might be the ones. And so they were able to clone those and study each one of them independently, and found one that it turns out had complement inhibition properties. It was a C5 complement inhibitor, essentially, coming directly from the tick. And then remarkably, that same protein had leukotriene B4 inhibition properties. So they found both properties in one protein that was, isolated from these thousands in these ticks. But yeah, so it was fascinating. I personally love the story because as a kid, I was always watching these nature shows and fascinated by, I don't know, poison dart frogs and geckos and whatever else that, we'll go on to some of these interesting, scientific discoveries that, we end up using in our lives. But I think with the tick story, as you have I would say foreshadowed, we definitely see some potential here to impact lives.
Yeah. I mean, it's always
in a positive way.
Sure. Yeah, in a positive way. That's right. Which is I don't know if irony is the right word for it, but it makes it all the more exciting. It's one thing, like you said, when you're a kid, if you're an inquisitive kid and interested at all in science, you like to think about the fact that that nature could contain the keys and the substances and the molecules to to improve health and cure disease, right? Like it's intriguing. It's a beautiful mystery to think about, but you wouldn't necessarily go directly to tick saliva. But you guys are working on this protein, and so the discovery happens, and I want to kind of work chronologically through how you go from discovery of this protein and its potential to a situation where you're actually building a clinical program and hoping to move forward with a therapeutic derived from and built on this protein. So I guess where would you start? Like from discovery, where would you start, and sort of the explanation of how the company has moved to the point that's at today, where I'm assuming synthesizing a version of this protein. We're no longer like hanging the tick's little mouth parts over a vial and squeezing, right? We're not doing that anymore.
No longer milking the ticks. That's right. That's right.
Yeah.
It's such a visual, isn't it?
Yeah.
I'll tell you a little bit more about that, because I do think it's interesting, and it goes to the kind of tenacity that teams like this have to make, major strides that, in fact, in the whole timeline for a biotech is really just the beginning. But there's significant work that went on. I described a little bit of the discovery process, but to bring us to what we call now nomacopan. But what is interesting is the number of steps that were between then and now. And you think about the first time that this team tried to make a recombinant protein from this. So they looked for, they identified the genetic sequence, and they worked to express that in a cell culture, and it was, and it worked. So that was a huge step, because if you think about what were they doing before to try to isolate the protein, they had to do these dissection sessions, and perhaps up to 12-hour days. Maybe they would get 200 ticks done. They would essentially, take their salivary glands, remove them, and then extract these proteins, and then of course have to purify them and isolate, what is now nomacopan. So that was an arduous and manual process, obviously not at all suitable for the kinds of things that we're doing now, clinical trials, and certainly ambitions for commercialization. So they knew they had to try to make a recombinant version. They did that. The protein behaved as they expected, had the same properties. So that was a huge success, but they were expressing that in kind of an insect cell, kind of a cell bank approach, and it just didn't work. I mean, it worked from it was producing the protein, but the yield was very, very, very low.
Yeah.
So they knew they could make a recombinant protein, but the yield wasn't anything close to what would be necessary for clinical studies. And then from there, as typical for many folks in our space, you take it step by step, trying to get yourself into a position where your manufacturing process is much more conventional. And so the company moved to a different cell expression and microbial environment, and that proved to be a much higher yield, so much closer to being something scalable. But even then, we had some ways to go. And if you look at something like nomacopan, I think one of the interesting things is, if you have some kind of a product that you think might be used in one small patient population, I think you can kind of get away with one certain cost structure. But the potential for nomacopan, we believe, is much broader across many disease areas. And so getting that manufacturing right has been really important to us. So I would say I could talk for hours about it, but I will tell you the bottom line is now it's pretty darn boring. The manufacturing, there's no milking of ticks, is very stereotypical biologics process where we're expressing the proteins, and we are making drug substance with a contract manufacturer, and we have another partner for drug product, and it is, again, utterly boring and just how we like it.
When you're striving to excel in a new arena, the best guides are the ones already doing it well. The Business of Biotech brings those voices forward to help new and emerging biopharmas turn their innovations, like mRNA and cell and gene therapies, into clinical realities. Tune in and subscribe for insights on hiring, regulatory, and other need-to-know topics for biopharma leaders. The podcast is brought to you in collaboration with Cytiva. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. Yeah, no, that's good. Yeah, but at this point in the juncture, at this juncture in the business, boring is definitely good.
Yes,
we've managed to have this conversation, without the context of the indications that you're pursuing. So I know I'm bouncing around quite a bit here, but I want to. I think it'd be helpful to have a conversation around the indications that you're pursuing and how that aligns with the manufacturing strategy. So let's talk about indications for a minute. What are you going after?
sure. So it is. I did mention that we think there's broad potential for nomacopan, and actually over the years, the company's done a lot of work in looking at different things and running different studies, exploring the possibilities. So we have all of that, but we've actually made a bit of a shift to focus and focus for nomacopan on a condition. I hesitate because it's a mouthful. The acronym is HSCT-TMA, but it's a hematopoietic stem cell transplant-related thrombotic microangiopathy. So you think, and we're focused on kids, pediatric with a severe version of this. And it's basically, you can think of a child that has like a stem cell transplant, bone marrow transplant, maybe because they have a underlying malignancy or other condition. And after that procedure, of course, they're battling whatever is the underlying condition. After that procedure, they can actually have this thrombotic microangiopathy, and it's a side effect, if you will. But unfortunately, it's one of these where in these kids with the severe form, it's 80% mortality rate.
Mhm.
And there's no approved therapy today. And so that's really where we've decided to focus our energies for nomacopan. It's rare. It's, perhaps you would call it ultra rare. I don't think there's any particular definition for ultra rare, but it is a small patient population, but very high unmet need. And so that's where we're focused and trying to move that forward as quickly as possible, knowing the urgency that is required.
Yeah. And where is that in the clinical continuum?
Yeah, we're in phase 3. So we have a phase 3 study going on. We have it, it's actually in 2 parts. It's a pretty interesting design. So we have 2 parts. So we're in what we call part A. And we do expect to have a readout in the first half of next year around that part A. And then we'll have a touch base with the regulators and move into part B, which is the final part of that study. So that's our lead program. We do have we've also prioritized a preclinical program, but it's with a pasylated version of nomacopan. This is a long-acting nomacopan, and that will be targeted to geographic atrophy. So in the eye, a different mode. It's an intravitreal injection, which is also very interesting. We think we've got a very interesting target product profile in particular related to the LTB4 inhibition.
Yeah. So given the rare to ultra rare state of the indication that you're pursuing with your phase 3 candidate right now, in nomacopan, what you mentioned scale-up. What is, so given the the rarity of the condition, what does scale-up look like in a situation like that for a business like this? Like, is it
it's straightforward, yes. So it's not a major challenge. It's very different from a large indication or for example, I talk about the preclinical pasylated nomacopan. That's a mass market. Geographic atrophy is a mass market kind of indication. Very, very different. For HSCT-TMA, it is we're already well positioned to be able to meet the needs at the scale that's necessary. And just to give you a perspective on the patient population, we're talking about 100 to 200 patients a year in the pediatrics in the U.S. That's just for a point of reference, but in the U.S. So yeah, absolutely, we have what we need in terms of batch size and scale and shelf life. And I think just to give you a flavor for scale change, yield change over the years. So we look at our first recombinant version that is, the version that basically the expression system that we use today compared to now. There, the team has already achieved like a 12x increase in yield. So that already sounds really good. But I always love the fun fact of if you compare where we are today to the way back in the days of the origins in the lab, one batch of nomacopan today contains the protein equivalent to about 200 million ticks.
Yeah. Wow. That's incredible. Yeah. That's coming a long way. Now,
that's coming a long way. I think the team should have some sort of an award for that, but I don't know what it is.
Absolutely. And that brings up a good point. So I want to make sure I'm clear on this. Nomacopan. So the proteins as found in nature in the tick, that is not nomacopan is like nomacopan
the
is
recombinant,
the recombinant version of that. So we wouldn't call that nomacopan in the tick.
No, it probably has some very, very long sequence name that I'm not sure has a catchy.
Not nearly as catchy as.
If I recall, it takes about 4 lines on a Word document.
Good to clarify. So I want to ask you a question about the strategy, the clinical strategy and the development strategy around those 2 indications that you mentioned. And I'm not going to attempt to pronounce the first one. That's super rare condition in pediatric. And then there's geographic atrophy. In any business outside of biopharma, probably in any business outside of biopharma, if I'm looking to develop a product that could have very specific appeal to a very small population versus that same product with a couple of twists that could have very massive appeal to a very large population, I'm probably going to chase the big one. first. What was this? What, there's probably some obvious strategic thinking behind pursuing the rare opportunity first, around regulatory, clinical speed, that kind of thing. But I want your perspective on that. What was sort of the business strategy around the prioritization of those indications?
Yeah, so you already hint at it a little bit, and when we look at when there are opportunities, and this is Rachelle's perspective here. So when there are opportunities, I think across a number of potential indications, I like to think about what is the highest unmet need, and what is the fastest path to a potential filing, to a potential approval. And when we look at HSCT-TMA, we know that there's no approved therapy. We know there's high unmet need. So it's I think easy for us to select that as an obvious thing that will have benefit if we bring it to market. And then there's, when you're working in rare diseases, I've been working in rare diseases for about a decade. It is, I think what we come to understand is that everybody in ecosystem is trying to move these programs forward. And so, looking at regulators, obviously they have a role. Their role includes making sure patients are safe and ensuring that, we've got controllable processes and all of these things that are incredibly important. But those same regulators also want to see these programs move forward if they are safe and efficacious. And so, being able to work together with the regulators to find the best path forward, there is some flexibility in those pathways. And so I think that's important to keep in mind as we're looking at how to move with urgency. Yes, we could go after big indications. As a small company, though, to take that on as your first bite at the apple doesn't necessarily work out. I think, the investment that's necessary and the scale and the scope of, forget about manufacturing, trying to reach all of the potential folks that you need to reach in the market, and the medical folks and everybody, to bring that to fruition. It's a giant undertaking. So I think going for what are the populations that need something most, where we can make the most impact, and then we can think later about whether or not we want to expand that. Now we have a little bit of a different situation and a little bit unique. A lot of times you have this pipeline in a product kind of a situation. It is difficult if you've got, well, I could go, and serve patients with rare diseases, or I could go after large conditions. Where we're a little bit different is that for our painstakingly nomacopan, it is actually a different asset. So it's a bit easier for us to bifurcate the two and think about those businesses, think about those as separate businesses.
Gotcha.
So, but yeah, we want to move with speed. The HSCT-TMA program is, again, there's a clear unmet need and a reason to believe, and a reason to, get up every morning and drive that program forward. We have other things that we can do in the rare disease space as well. And I think, as a small company moving closer and closer to hopefully eventual commercialization, you want to build bit by bit and make sure that you've got the right capabilities in place and so on. And so we have that opportunity.
Yeah. Are there any other, any other candidates coming on the heels of these two indications that you discussed? Any other kind of pipeline updates beyond those two that you can share?
We are trying really hard to stay focused, which is incredibly difficult. It's hard to believe. It is such a good problem to have, but it definitely requires discipline. So our objective right now is to stay focused on those. But if you were to look at a little bit of history around Akari, we've absolutely looked at other indications, and those are still potential in the future. We've just kind of put those away so we could focus our resources on the lead program. And I think it's much easier once you have if you can reach a marketing authorization with the lead assets, it's much easier than to pivot to additional indications and investments in those. It's a little bit, and it's no guarantee that it's de-risked, but it's a little bit of a different profile, and you can build on that first success.
Yeah, very good.
Yeah.
What haven't I asked you, Rachelle? What haven't I asked you that I should have? What are some big elements of the story that perhaps I'm missing?
Gosh, why haven't you asked?
You know,
I don't know. I think you're pretty thorough. I think, for us, probably the big thing in terms of the difference with nomacopan is this bispecific nature. And, I think that's one piece that is, it is quite interesting because we've got two mechanisms of action. One is the complement C5 inhibition, which is well known and understood.
Mm-hmm.
And the other is this leukotriene B4, which is part of the leukotriene pathway. LTB4 itself is a pro-inflammatory. And so when we think about, inhibiting both of those pathways, what we're doing is we're trying to address inflammation from two different pathways, cell damage from two pathways. And, a lot of that results in organ damage, sometimes, unfortunately, death. And, we're working in the area of autoimmune and inflammatory diseases. These can be complex, and there are a lot of interrelationships we know from a biological standpoint in these systems. And so being able to address that inflammation and cell damage from the two pathways, we do believe is promising. So that's for us something that, we believe is different about nomacopan and is quite exciting in terms of, hopefully being able to, result in better outcomes for patients.
Yeah. And as you mentioned, you gave us the update on where you are clinically, and I understand that you recently extended your cash runway a bit too. So you put yourself on some pretty sound financial footing to move forward.
Yeah, we did. We just finished a financing and are very pleased because that really gets us into our next milestone, which is this data readout in the first half of 23. Mm-hmm. And so we'll go from there. But that's definitely exciting. It's a challenging time out there, and I say, my thoughts are with all of those who are working on finding the financing and the support they need for some pretty exciting programs in our industry, but is definitely challenging and uncertain. And every time we think it's going to get better, it hasn't quite achieved that yet. So we will, I think we'll remain open to watching what's going on, but at least for Akari, we've got ourselves secured through data. So that's a pretty exciting development.
Exciting. Yeah. Congratulations on that. It's like
thank you.
It's always a big accomplishment, but it's obviously a much bigger accomplishment in a very challenged capital market like we're seeing right now. Obviously, I don't expect you to share the silver bullet in your pitch deck that made it work, but any advice for those who are kind of struggling through this right now? Because to your point, it is different than it was 4 years ago. It's different than it was 2 years ago. Like we were talking before the show, 2 years ago, it was, oh my God, how am I going to make any money? How am I going to raise any money when I can't get on an airplane or stay in a hotel or go to a conference? And, Zoom saves the day. Everybody finds it relatively still at that point. I don't want to say easy. It's never easy, but relatively available, right? Capital. And then here we are 2 years later, coming off of that challenge, faced by a whole new one, high interest rates and very discerning investors. I don't know. So, maybe your advice to the folks who are out there on that trail right now is just fortitude.
Fortitude.
Keep your heart
I
in
say
it.
Stick with it. Know your boundaries and, but be open because, I think a lot of what's important is that we continue to have the financing to move the programs forward and hopefully are driving value by achieving those milestones in the future. But fortitude's a big part of it. Yeah. Keep at it. I don't know. Everybody's in the same boat, and so I think just being able to secure what you need to get you to that next place is pretty important. So,
Yep.
yeah,
Data.
I have no magical solution. We're definitely not magical unicorns, but I think everybody is finding their own path, and it's interesting to watch the different options. You see more in terms of debt financing, royalty streams, and all of these things. And I think people are being creative as they need to meet the times.
Yeah. Yeah.
Yeah.
Well, and like everyone always says, data earns money. So we'll be keeping an eye on the data coming out of Akari Therapeutics. It's an exciting, mind-boggling, beautiful thing that you're doing. The origin story is amazing. I still hate ticks. And by the way, I was going to mention that if clinical supply, as you move towards geographic atrophy, and you need to produce more, you need that scallop actually becomes like a big scallop. You need more clinical supply. Northwest Pennsylvania, target-rich environment. If you need to go back to the original source, I'm just going to throw that out there. I know.
Will you take me tick hunting? Is that what you're saying?
All you got to do, you take a walk through the woods, you get home, and you just pick them off your
Just
clothing.
brush them off.
It's true. I know it's a far less efficient means of gathering the protein than recombinant technology that Akari's developed, but if it comes to that, target-rich environment. So.
I think my manufacturing leader is probably not going to take our basket of ticks, but I will keep my mind open.
Well,
thank you for the offer.
Yeah. Well, it stands. We can tick hunt in any event, whether they're useful or not. But I thank you for coming on. There. Yeah, I thank you for coming on and sharing the story with us. It's great to catch up again. As I said, we'll be watching the clinical progress and hoping for the best, because it's always a more beautiful thing when it's when you can make lemons, make lemonade out of lemons, right? Make something good out of something that is otherwise.
That's right.
otherwise looked at as far from desirable. So.
That's right. Well, we look forward to sharing the progress. So thank you. Thanks for the discussion today. It was great. Good to talk with you. And hopefully you'll like ticks just a little bit more next year.
Indeed. Thanks, Rachelle.
All right. Thanks, Matt.
That's Akari Therapeutics President and CEO Rachelle Jacques, whose disdain for ticks is far lesser than mine. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva. And if you visit Cytiva's Emerging Biotech Accelerator at cytiva.com/emerging biotech, I promise you won't be ticked off by what you find there. Visit us at bioprocessonline.com. And if you like what you're hearing here on the Business of Biotech, subscribe, share us with a colleague, leave us a review. And as always, thanks for listening.